then they come down and form a complex that becomes another enzyme. So initially form a C3 convertase that again convert into C5 convertase. They will convert this C5b and form the membrane attack complex. Similarly, there is in the mannose binding lectin pathway. This is the same. This is your bacterial surfaces. Okay, so the bacterial surfaces, you can see on the bacterial surfaces, this bacterial surfaces carbohydrate, the lectin is going to bind over here. After binding the lectin, they will activate again this C4b and 2a. There is no this component in the mannose binding pathway, so they are part of your innate immune system. There is no requirement of antibody to get activated. So there is a bacterial microbial surface, lectin is going to bind. After binding, they will activate this two protein, they will cleavage this C4b and C4 and C2, they will form this C4b and 2a complex. This is called C3 convertase. This will cleavage the C3, they will again C3b will be incorporated over here. They form the C5 convertase, that is the 4b 2a 3b C5 convertase. This C5b convertase will cleavage this C5 and this C5b 6 7 8 9 will form the membrane attack complex. So classical pathway, there is requirement of antigen antibody and C1 protein to get activated. In case of mannose binding lectin pathway, this C1 is not required. You just require this lectin to bind with the microbial surfaces and then this get activated while they form the C3 convertase. After forming the C3 convertase, C3 will be cleavage, form the C5 convertase and C5 will again cleavate and form this membrane attack complex. So in mannose binding protein, you don't need to have this classical effect, classical antibody requirement and complement C1 to be activated. So they are the part of your innate immune system. There is another pathway which is called the alternative pathway. This also does not require antibody to get activated. Actually, this is the microbial surfaces, okay, bacterial surfaces where this C3b goes and get attached. After attaching, there are the certain factor like factor B and factor D which comes and then they helps this to get activated. So once factor B and D comes, this factor B will go and attach over there. So this 3b C3b and factor B, that is big B and small b, will make and form a C3 convertase. You have to remember the initial C3 convertase was C4b 2a. So C4b C2a. So this is C3 convertase for classical pathway and mannose binding lectin pathway. But in case of alternative pathway, the C3 convertase is not as similar to that of the classical or mannose binding lectin pathway. That is totally different, and the different is that C3b and there is a big B and small b. So C3 convertase of your alternative pathway is different than the C3 convertase of your classical or mannose binding lectin pathway. Okay, so this becomes your now C3 convertase. Why? Because this C3b has come on the bacterial surfaces, factor B has came, so there will be a C3b, big B and small b. This is C3 convertase. This will cleavate this C3 and form this C3b, C3a. So we know this B will go and bind, so there will be a C3b, big B, small b and C3b. This will come and attach. So C3b, big B and this are from the above only, and this has come and again attached. So C3b, big B, small b and C3b, these all are now become C5 convertase. So now you have C5 convertase. So this C5 convertase will now cleavage this C5 and then form the membrane attack complex. So alternative pathway, or say lectin mannose binding pathway. So what is the main point? The lectin mannose pathway or a classical pathway. The classical pathway, there was antigen antibody and this C1 protein which was not required in mannose binding protein pathway. In mannose binding pathway, this was reactivation of C2, 4 and 2 which was not required in the alternative pathway. Alternative pathway direct activates C3b protein, which in the presence of factor B and factor D, they form a C3 convertase which is new, not similar to that of the classical or MBL pathway, and this will form again cleavage this C3 and form the C5 convertase which will again cleavage the C5 and form the membrane attack complex. So in alternative pathway, there is not requirement of this C4 or C2 as well. Also not requirement of C1. Okay, so hope you have understood all the pathway. This pathway are important, but you have to remember the end product of each and every pathway is this membrane attack complex, which helps to killing the pathogens, which helps to kill the bacteria or say pathogens. Okay. So this is the summary of the same thing. The classical pathway, that is antigen antibody complex, they require this C1QRS that activates C4 and C2. This form the C3 convertase. This C3 convertase cleaves the C3, and C3A goes over here, form the C3B, get attached and form the C5 convertase, and then form the membrane attack complex, and this kills the bacteria. They have the C3B, which helps in the opsonization as well as removal of the immune complexes, and the byproduct that has relieved in the environment that will cause the inflammation and phagocytic requirement. Okay, the similar things apply for this mannan binding lectin pathway. This mannose binding lectin bind the mannose on the pathogen surface. This MBL, MASP1 and MSP2 helps to cleavage and activate this C2 and C4. They will form this C3 convertase. This C3 convertase will again cleavage this C3. A will release over in the environment. B will form here. This will form the C5 convertase. This will again 5A is released, and 5B will come here and form this membrane attack complex and kill the pathogen. They even helps to form this phagocytosis, opsonization and removal of the immune complex, and mediate the inflammatory mediators and phagocytic recruitment. There is another pathway called the alternative pathway that has the pathogen surfaces, which has this C3, and they require this factor B and factor D, by which they will get activated. So there is no requirement of C2, C4 or C1. Only the C3 will be activated in the form of B and D factor, and they form the C3 convertase. But the C3 convertase will be C3B, big B, small b. This is the C3 convertase. They will cleavage the C3, and another C3B will be added. This will be now C5 convertase, and this C5 convertase will convert the cleave the C5 and form this C5B, 6, 7, 8, 9 membrane attack complex, and C5A will release in the environment, and that will cause the anaphylotoxin. So hope you have understand this chapter in detail. Now let's move about their function. The classical pathway, we have known these are the depend on the antigen antibody complex. They are actually activated by this immunoglobulin M and G. So M and G is required, but except G4, immunoglobulin G4 will not activate this classical pathway. They involves the activation of C1Q, R and S bind to the Fc portion of the immunoglobulin G and M and require calcium for the activation. So let's see over here. This is a classical pathway. When there is an antigen, antibody will form, then antigen antibody reaction will activate your C1Q, this C1, then they activate R and S. In the presence of calcium, they will cleavage this C4 and C2, then they will form this C4B, 2A. The 2A has, B has the exception, B will go away, A will has a bigger molecule, so they will attach, and this convertase will form as the C4B 2A, that is the C3 convertase. So once C3 convertase is formed, this will cleavate the C3, and C3A will release, B will bind. This all component is now called the, initially this was the C3 convertase. Now this whole will become, now if they have cleavage the C3, then form the C5 convertase, and this C5 convertase will cleavage the C5. C5 will be cleaved, and C5 will be attached, then 5, 6 and 7 will come, then 8 will come, then 9 will come, and this will form the, this all is membrane attack complex. This all membrane attack complex is formed that has now cleaved your bacterial cell, cause the lysis in the bacterial cell and kill that bacterial cell. So this is your membrane attack complex. Looks like the missile because they enter inside the cell, form a pore, and then kill that bacteria. So you have understood this whole the pathway and the activation product. So alternative pathway we have discussed. This is actually bypass. There is no requirement of this C1, 4 or 2. It does not require antigen binding protein, does not wait for antibody to be formed, and acts synergistically with the classical pathway, also known as the proprietary pathway. This is usually activated by the product of the microorganism like endotoxin. Also other complexes can activate these things. This pathway is actually microbes. You can see the factor B, D and P is there. They cleavage the C3B. They form the C3B convertase and cause the C3B, then again goes to the C5 and form the C5, cleavage that C5, and then form the membrane attack complex. We have no, then is the alternative pathway, that is find C3B finds an activator membrane. This is stable C5 convergence of the alternative pathway. Lytic pathway, we have discussed about this initially also, the same thing is there. Okay, then lectin pathway. In the lectin pathway, just you need mannose binding lectin. So lectin of our cell, our body would bind with the mannose of your bacteria, and then they get activated, and then they activate. After activation, they form the same membrane attack complex and kill the bacteria. So lectin is enough to start this complement system to get activated on the microbial surface. Okay, so now we have come to this. We have understood about this. We have discussed in detail about how this one after another protein get activated. We have talked about the function. This as the one is the opsonization that enhance the phagocytosis and kill the bacteria. Then there is the chemotaxis that bring all this bacterial or phagocytic cell there and attract and produce the inflammation. There is the anaphylatoxin that cause the degradation of the mast cell, bind directly to the smooth muscle of the bronchioles, cause bronchospasm, and also they kills the cell, which is the main function of the membrane attack complex, that is disrupt the membrane and entry of the water and electrolytes into the cell of any bacteria or pathogen and kill that bacteria.